nucleus where the chromosomes are working. So this string will represent the nuclear membrane. And within that is the nucleus with the chromosomes. Each one of these represents a different chromosome. They all have the chromosome arms where the genes and the alleles reside, and centromeres in the middle where the mitotic apparatus connects. And since this is a diploid cell, there's 2 copies of the long chromosome and 2 copies of the short chromosome. We'll call the long one chromosome 1 and the short one chromosome 2. And they're different colors to represent their different origins. That is, the purple ones will be the paternal chromosomes. That is, this copy of chromosome 1 and this copy of chromosome 2 came in the sperm cell from the father. This copy of chromosome 1 and this copy of chromosome 2 came in the egg cell from the mother. So this is an interphase resting cell with 2 copies of each chromosome about to undergo mitosis. Now, the first step in mitosis is that each of the chromosomes is exactly duplicated. And the exact duplicates are linked together at the centromeres. And the cell also grows during this period of time. The nucleus will get larger. And you'll notice that each of the copies joined together are identical copies. 2 copies of the maternal chromosome 2, 2 copies of the paternal chromosome 2. Again, joined at the centromere. And these are called sister chromatids. This is one sister chromatid, another sister chromatid. And so these are identical in every, each of these 2 is identical in every respect. The chromosomes here to here are identical. Every gene, every allele is the same on both arms. Whereas these chromosomes, the purple to the white, are homologous. That is, the genes are all in the same order on both of these, but they may have different alleles having come from the mother or the father. The next step in mitosis is all the chromosomes line up along the midline of the cell and prepare to divide. At this point, mitotic apparatus attaches itself. These are called spindle fibers. They're made of microtubules. The cell assembles them onto each of the chromosomes, onto each of the centromeres of each of the chromosomes. And this is what is called metaphase of mitosis, with all the chromosomes lined up. They then begin to pull apart as the cell continues through mitosis. They separate at the centromeres, and the spindle fibers pull the sister chromatids apart, the identical copies apart, so that one copy of each chromosome goes to each of the daughter cells. And as the chromosome arms are dragged through the medium of the cell, they bend back like this. Gradually they pull apart, and then the nuclear membrane comes up between them and divides off into 2 cells. The mitotic apparatus disengages. Now at the end of mitosis, you end up with 2 cells, one here and one here, with identical copies of the chromosomes. That is, they each have a copy of paternal chromosome 2, maternal chromosome 2, maternal chromosome 1, paternal chromosome 1. And they're identical copies of the original ones. Mitosis is complete, and these can go through a further round of mitosis after this.